This is a production of Cornell University.
Okay, good. Thank Kathy for that wonderful introduction. It's really a bit emotional when I get this invitation. It's a great honor to be invited by your alma mater. And I remember last time I gave a seminar in that room was for my B exam and for my dissertation. And that was 17 years ago. So I think by that order, probably I can consider this as my C exam. And okay, so today I'm going to, oh yes, I want to thank Kathy and Gary and also Gillian for giving me this opportunity. That's okay. So today I'm going to talk about the genetic diversity of ergot fungi at species and population level, and also in terms of production of ergot alkaloids. So I'm going to give a quick historical review of ergot taxonomy in pre-molecular era and also molecular era, and then showcase a bit research results from our population level study. And towards the end, I want also introduce some research we collaborated with other labs in terms of the variation of ergot alkaloid production and also at the gene level. So before I start, I like to give a quick life cycle, go through the quick life cycle, and so that everybody get a bit background. So the black grains on this broom grass, can you see my like a mouse moving?
Yeah, we can.
Okay, that's good. Okay, so these black grains are the resting body. Sorry, this black green are the resting body of the fungus we call the ergot fungus, and they during the fall they will drop on the ground and over winter, and next spring they will germinate, produce these drumstick like little structures. They are sexual reproducing structure, and the inside of this big head are the sexual reproducing protogules, which are ascospores arranged in a special order. And when the time comes, and they will disperse into the air and carried by the wind to infect a flower, specifically only the flower of grasses or cereal crops. And the germinated spore, the hyphae will keep growing towards the bottom of the ovary. And then, interestingly, they will travel back and not further to the plant, but travel back, grow inside of this ovary, and keep growing and develop, and eventually the seed, the space within the seed, and all filled with the asexual spores Canadia and wrapped with honeydew, and those honeydew will ooze out and get stick on the insect legs, and when the insect fly onto another plant get infected, and this cycle will repeat during the planting and the flowering season. And towards the end of the season, the inside of the seed will be fully packed with mycelia, and the mycelia will be hardened and also pigmented and become like this resting body or ergot. And then the cycle will repeat again. So ergot, according to the literature, the earliest record of ergot, it's in this German book. That's the earliest I can trace out. Maybe Gary or Kathy had more information, but anyway, in this literature, they called this recorded ergot as long black nails between rye grains, and at that time, they all considered ergot as abnormal grains. So there were all kinds of hypotheses. One hypothesis said that they were sun-baked grains, or the excessive formation of sap, or just simply mutation of grains. Until 1771, Geoffroy, a French pharmacist, considered ergot actually is a separate plant from its host plant. And remember, at that time, fungi were considered as a plant. And about 40 years later, Digando agreed his hypothesis and assigned a name to ergot, Sclerotium clavus. And another 40 years later, Tulassen discovered the sexual stage of ergot, and he erected a genus as claviceps. And also he considered the species he discovered is the same species with the one phrase called a name called Sphaeria purpurea. That's why he called this name, he renamed the fungus as Claviceps purpurea, and also noted Dicando's Sclerotium clavus is the synonym of the Claviceps purpurea, the fungus he's working on. And in addition, he also recognized two other species, and C. microcephala and C. nigricans, and based on the morphology and the biology, such as C. microcephala, it produces this very, as the arrow showed here, very long flexures stipes and very soft little head. And also biologically, microcephala, the sclerotia can germinate immediately on the crop, and the sclerotia does not need to drop on the ground and after the freeze treatment and germinate again, and which is most of the species will need that. And in terms of nigricans, this is on the sedges, and the morphologically, according to Tulassen, the paracesia arranged more distantly than other species. And after Tulassen, ergot species were described here and there. And this is the list I summarized from Index Fungorum and the Microbank. And I might miss a few, but I will say in overall, it's not a long list. And also a side note here, the taxa in species in red font were classified to other genera based on molecular taxonomy later. And until 1940s, 1940 to 1950s, London from Australia, he did extensive work on ergot. He described some new species from Australia and also evaluated 25 existing species. And in addition, he also evaluated the usefulness of morphological characters, such as, according to him, the shape and the color of sclerotia are informative, but not the size. And in terms of stromata, he thinks the color and the stipe, the color of stipe and the capitula are informative, but not the length of the stipe. He said the length will keep growing when the stromata grow in mature. So it's not very informative. And also he did say the placement of paracesia is informative. And also he mentioned in his dissertation some specific characters, such as a collar around the base of a capitula, a white ring, or a loose hyphae around the capitula. Unfortunately, he didn't show any picture of those characters, and we will just have to pay attention to that when we do our taxonomy. And also in terms of the Canadia, and he agreed that the shape and the size of Canadia are different between species. However, they are also variable within species. So it can be useful, but to a certain extent. Lovelace in between 1950 to 1970s did more careful work in terms of the shape and the size of Canadia. He studied the Canadia collected from 34 grasses, and in Rhodesia. Rhodesia is a place nowadays close to equivalent to Zimbabwe. And what he did is he randomly picked 25 spores and grafted them with special tools, grafted them and outlined them and separated, categorized them to 13 groups. And then he was able to associate 6 of the groups to already described species, such as this group 2, say, Paspali, and 6 species. And also he predicted there should be another 7 species in this area. And what interesting is after many years, several species were described in South Africa, the area close by. And it turned out, the evaluation turns out the Canadians did match with his prediction. And it's kind of amazing. And another name people can encounter when you study taxonomy for ergot is Tanda, and from Japan. He is a person, focused on varieties. He did describe a few species, but characterized a whole array of varieties. And he did based on very, very careful study, such as this one. I show a plate from his publication. He studied the shape, size of the sclerotia and stromata, paracesia, Canadian shape and size, and even the honeydew, sugar content and alkaloid content. Did very detailed comparison of the ergot from different hosts. And the one from phalaris, he did noted that this one had a significantly larger Canadia, but he was not convinced it's a new species. He just called it variety, like Subpopulations variety phalaridis. And actually, based on modern molecular taxonomy, this turned out to be a new species and described later by Pazotova group called humidiphylla. And in the same way, many of his varieties need evaluated. Some of them might represent a new species, some may be just at variety level. And the sugar content and the usefulness for taxonomy was studied by Moore and Hancock in 1975. And they were able to recognize a few taxonomy groups, and they have different content, such as the group A, the species in group A are rich in monosaccharides. However, in the species in group 2 are rich in sugar alcohol. And also there are also some species just mixed with like he call intermediate. And compared to sugar content, more studies were attracted for ergot content study because of their significant impact to agriculture, because they are toxic, right? And also because of their pharmaceutical potentials. As I indicated, color-coded here, like different species contain different sets of ergot alkaloid. And I will talk about this topic later in my presentation. So move on to molecular error. The group in Czech Republic led by Pazotova did a pioneer work for ergot taxonomy. They were able to characterize the different species with RAPT marker and the ITS1 markers. And as I show the picture here, different species had showed different profiles. And in addition, they also developed this new media, high sugar content media, they call it T2 media. And usually claviceps on the PDA media will grow very fluffy and white and difficult to tell apart the species. However, on this media, you can see different species will grow more compact and show different texture and also different hint of colors, which can be used for differentiate species. And also on this media, the claviceps can tend to produce more Canadia, and this gave them opportunity to observe more characteristics of the Canadia, and such as the presence or absence of macros Canadia, as I show picture on the left, and this oval-shaped small size Canadia. Sorry. And the presence and absence and also the shape can be different between different species. And another feature is they call it secondary Canadia. And what happened is the Canadia, when the Canadia land on the media, they will start to germinate and produce germ tubes. And the germ tube will produce another Canadia. And that Canadian can produce germ tube and produce another Canadia again. So they call that microcycle. And the different species, these times of microcycle different. Some species just once, some species maybe 2 or 3 times. And the use multi-gene phylogeny to differentiate claviceps species started with a US group, and they were able to separate the C. purpurea sensu-lato into 3 groups, and these 3 groups are actually called consistent with the 3 chemoraces identified by Pathology group, and they also indicated these 3 groups are associated with ecological niches, indicating a kind of ecological speciation. Until 2015, Pathology group conducted a formal taxonomy treatment for those 3 groups and gave each of them a name. And in addition, they also found the third lineage, and they named it C. arudinis. And as a side note here, this concept of arudinis is equivalent to C. macrocephala tulascens concept. However, the type of C. macrocephala had some issue, so they renamed the fungus as arudinis. And if you are interested, anybody interested in the details, and can look this publication, and I'm not going to too much detail. And for this clade, Pathology group called it C. humidifera. Basically, it is the same taxa. They considered it's the same taxa with the one Tanda described as C. purpurea variety phaloridis. And what's interesting is our follow-up study just recently between me and EG and also Miroslav. We discovered that actually this clade actually is different clade, different species with the Tanda's variety, and it's a bit complicated because of the type specimen, the designation of this type had some issue. And so in a short word, this taxa right now, correct name, we gave it a new name called C. bavariensis, and the real C. humidifera actually is restricted in Japan. And yeah, if you for more detail, you can look into this publication, and it's going to publish soon, next week. This is the preliminary PDF file. And another advance in the taxonomy of claviceps is another comprehensive multi-gene phylogenetic analysis by this Pathology group led by Miroslav Kolaria. And it's kind of sad note that the Dr. Pathology passed away, and I think just before after 2015 paper published around that time. So it's kind of very sad. And Miroslav, the guy stand here, he is the lead right now, lead that group. And in this paper, they separated the genus Claviceps into 4 sections, and also studied their alkaloid production and host correlation, and very comprehensive, excellent work. And also in a supplementary table, and they were kind of shy, and they listed all the existing species names, and they kind of gave a preliminary evaluation of those names, whether it's valid or not valid. It's set a very good foundation for a monograph. But unfortunately, it's in a supplementary table, and if you don't pay attention, you barely notice that. And here in Canada, in our National Mycological Herbarium, there are about 700 Claviceps species, and 90% of them labeled as Claviceps purpurea. And in addition, there are a few more species described in record. I think a part of the reason probably because Claviceps has never been seriously studied by a taxonomist in Canada. And that's why since 2016, my lab started working on this genetic diversity of this species in Canada. And we, with the help of all the friends and the colleagues and the organizations, and we got a big got a big collection from the agricultural and the non-agricultural area. We did a multi-gene phylogeny. This is a multi-gene phylogeny, and on this phylogeny, phylogenetic trees, all the 3 species discovered by Padotova group recovered here. You can see Spartina, Humidifera, and Aerodinici. And in addition, there are also 4 other lineages stand out, and they represented a new lineage, and we described them as a species, such as this one is closely related with Spartina. We named it as Ripicola. It was collected like around the river banks and sand dunes and lake banks, and like a semi-wet area habitat, and it's quite common. And we collected a bunch of species from Quebec, from Alberta, from different areas, Manitoba, from different areas. And this one, we got only 2 collections actually. On this tree, only 1 collection. Later on, we collect another collection. And this one was collected from Newfoundland, from salt marsh. And it's on Agrostis. And the character of this species, you can see on this culture petri dish, the culture place, and they produce very, very compact Canadia the colony. And you can see also with this root system like the structure is very unique from others. And 2 other species, C. cubensis, collected from Quebec, and characterized by this oval-shaped Canadia. And it's not microcanadia, it's bigger than microcanadia. And C. occidentalis is also very interesting. We only have the culture from our culture collection. And originally the specimen was collected in Alberta in 1950s. And I asked my friends and colleagues to go back to the collection location, original location, try to collect it again, and we were not able to collect it so far. So hopefully we can just keep watching it and collect it. But meanwhile, it also tells us how important our cultural collection is. So there might be some new species embedded there. So add to the new species we described, this is the updated, up-to-date list in Canada. And I believe there are more new species and waiting to discovering. And so now I'm going to go to talk a little about our population level study. And so let's go back to this big phylogeny, and a question would ask is for this big clade, and it's basically a bush here. And we will ask, is it truly just a bush because of the pan-mixed population, because of the, the recombination, frequent recombination between the species, so the exchange of genetic material, that's why there is no separation, or whether there are actually some kind of subdivision. For that phylogenetic tree, and the strict phylogenetic analysis, and it's based on the assumption the relationships among the taxa are tree-shaped, are bifurcate, and it's not good for the inferring the relationship among the taxa with the horizontal gene exchange. And instead, a network-based analytical tools and recently developed take consideration of that reticulate relationship, and will be better for that system. So with that in our mind, we looked into some of those kind of analytical method. So we analyzed the same data, the big clade with multigene data with a split tree. So split tree basically separated 146 haplotypes from 156 sample into 3 different clade. Yeah, as I show here, this network, and GC stand for genetic cluster, and GC, you can see GC1 is more deeply separated from GC2 and GC3. And GC2 and GC3 look like more close. And it's kind of nice to see a pattern, but we were wondering whether this is a consistent pattern or just a coincidence. So to confirm that, we looked into other methods such as BEAST, and also the principal coordinate analysis. And from this graph, BEAST, and also this graph, and you can all tell GC1 is more distantly separated from GC2 and GC3. GC2 and GC3, there is a little bit intermingle and probably indicate their recent separation, or maybe just error, and we don't know. And we also did some population statistic test, and so I don't have, you don't need to understand each of these numbers values indicate, but basically these values indicate there are very strong suggestion of population subdivision, and with limited gene flow among the populations. And with that, and we also ask a question whether this separation correlated with the host, with the geographic areas. And so we mapped those different genotype, all the samples on this map. And you can see it's not correlated with the geographic location. We separated the geographic, like West Canada, East Canada, and West US. Each of these regions contain 3 genetic clusters. And this is the graph, and a similar story, although with different portion of different genetic clusters. And in addition, then we ask whether it's correlated with host, and we separated the host also to 3 groups, like crop, tritici, mainly tritici crops, and non-tritici crops elements, this, and also broomy. And by this separation, you can also see on these 3 crop, these 3 host groups also contain each of those 3 genetic clusters with different proportions. But I think in take-home message is there are 3 subpopulations between Canada and West US. However, those subdivisions is not strictly correlated with the geographic location or the host separation. And here I would like to acknowledge my technician Parvash Shokohi, and also the co-op students, and they developed all this data. And students come and goes, and they learn the techniques, but meanwhile also have contributed on to the data development. So now I like to switch gear, talk about the variations in terms of alkaloid production and also at the gene level. So alkaloids are mycotoxin contained in mainly in ergot bodies, and by the complexity, they were classified into different classes. As I showed here in this graph, they include the simple clavins as these green colors like cyanoclavin, agroclavin, alimoclavin produced in early steps of the pathway. And also the ergopeptides, the most complex ergoalkaloids produced at the end product. And in between, there are dihydroergoalkaloids and LA-amides. So since the ergoalkaloid was isolated, I think it's 1950s, and for the first time, there are more and more alkaloids compounds were isolated from different species with different techniques. There are several good review papers as I listed here. And if you're interested, you can always look into these references. So here, I'm not going to like systematically go review all this. I will just like highlight a few point research results and from our collaborative work, and to show the variations of ergoalkaloid production at a different taxonomy level and also at the gene level, at section level, and as indicated by Petrova 2018. And the section Pusili, C section Pusili mainly produce the simple clavins and maximum some intermediate ergoalkaloids, and also section paspalorum is also like some simple and intermediate ones. The complex ergopeptides is exclusively produced by section claviceps. And for section C, citrina, for species C citrina, and the only number in section citrini does not produce any ergoalkaloid. That's the section level. So for the species in the section claviceps, they may contain different set of ergopeptide, such as the new species we described. C. ripicola mainly contained ergocreptide and its epimers. For C. cubacensis, in addition, ergocreptide also has ergocin. And but for C. perihumidifera, it's mainly composed by ergocreptide. And so Dave Overy is our mycologist, but also a chemist, and all our chemistry related work is conducted in his lab. And so if you have any questions in terms of chemistry, so you can contact him with this email. And another study by a Norwegian group, they also confirmed the ergot alkaloid profile, not only ergot alkaloid, but also other indoor alkaloids profile among these three species are different. And we also looked into the intraspecific variation. A master student, Carmen, did an experiment, and with she's also Dave's student, but with collaboration with Jim Manzi, research scientist in Modern Research Center, Plant Pathologists, and also my lab. And what we did is we selected some isolates, 8 isolates of Purpurea, inoculated them on 4 different crops. And then when the crop cereal, when the grains mature, we harvest the sclerotia. And then separate the sclerotia to 2 parts. One part used for profile of alkaloids, and another, or 1 part for the half for the identity, for confirm their identity, and another half for the profile, ergot alkaloid profile. And this is the result. On the left is from untargeted metabolomic profile, and you can clearly see this is from wheat, the sclerotia from wheat. You can clearly see they are separated to 2 classes, the red and blue. And the same pattern is consistent from for the sclerotia from other hosts, other 3 hosts. And on the right is ergot alkaloid profile, and you can also look at the pattern, and it's very clear. The red, this column, the red columns are the class 1, and the blue is class 2, and you see they have different patterns of mass features. Notably, this in these boxes, you see some mass features only present in class 2, but some only in class 1. And if you want to know more detail, refer to this publication. So another question we ask, remember for the population, we found that this subdivision, and we can consider GC1, GC3 as one big population. We were wondering whether this subdivision actually correlated somehow consistent with these chemo classes. And unfortunately, the answer is no. And it would be interesting to understand why it's no, and what are the mechanisms underneath this separation. And in order to understand the variation at the gene level, and also what kind of mechanism at a gene level might result to the separation of the chemo classes, and even also the population classification. And we initiated our genome project with collaboration with Miroslav, a Czech Republican group, and Kirk Brothers. And he was a professor in State University, Colorado, but now he's in USDA. And also with our AFC BioMob work package 2 is a part of a big project, like this work package led by Jeremy, our mycologist in our group. And we sequenced 53 Illumina genomes with Illumina platform, and also a few with Nanopore. And Steve is a student of Kirk, and he focused on comparative genomics, and we focused on the divergence of yeast genes. And here I will just only talk about these yeast genes. Wendy is our bioinformatician, helped with the pipeline and of the assembling and the gene cluster extraction, and Dr. Nguyen also supervised Wendy and help us. And he's also a research scientist, our mycologist. So based on previous study, ergot alkaloid in Claviceps purpurea, there are 12 genes, including 3 NRPS genes, LPSA, B, and C. LPSA had 2 copies, and they are super long, and it's not possible based on Illumina sequences to grab the whole gene. So that is why we did a nanopore. But for other 11 genes, we were able to extract them from Illumina sequence. And this is the results from the Illumina sequence. And it's kind of busy. The take-home message, yeah, this is a different species of the Claviceps, and this from across is a different genes. And basically the take-home message is between species, and we found the gene number and the gene copy polymorphism. And also for the section, in section Claviceps, it's more consistent in terms of gene copy, relatively consistent in terms of gene number and gene copy. For Pusilla, it's more variable, such as C. africana, we detected 8 genes, but Maximans none. And this actually is very nicely correlated with their production. And this is a reminder from the Petrova paper. And on this tree, you can see Maximans produce none, but Africana produce some dihydroalkaloid, and even some simple alkaloids produce some. But in other cases, it's not that consistent. Such as Pusilla and Soji, we detected 8 to 9 genes. For Pusilla, most of the genes are kind of non-functioning, look like non-functioning, so it's understandable. But for Soji, most of the genes look functioning. And however, in this, in their chart, and no ergot alkaloid production is detected. And this also worth to more investigation. What's more? This is within species. Between the strains, among the strains, all the strains, the variation among the strains basically is some strains contain the second copy of ES-E, ES-F, or even third copy of ES-F, and also a few strains doesn't have the second copy is ES-H. In terms of the gene copy polymorphism. And we also looked into the DNA sequence variation across the different genes. And we looked at the gene diversity and gene divergence across the different genes. And what we evaluated by 3 parameters: nucleotide diversity and tree-based diversity, and divergence from center of tree. And so what we found very interesting is the genes in early and late step of pathways are more diverged, have more diversity than the genes in the center. And this is very interesting because it's kind of resemble this model called hourglass model in ontogeny. And this ontogeny hourglass model was proposed in 1990s, and they indicated the one-fifth developer and in early and later stages are more divergent and not in the center. And so, and this hypothesis, this model, there has been a heated debate and between this model and also other models. There are also early conservative model indicated the early steps should be more conserved, the later steps are more diverged. And also there are also other models, I'm not going in detail. And now, And so, and there are vehement debate between which model it's, it's got more evidence. And recently, some genomic work did provided more evidence for this hourglass model. And our question here is, this model with this shape we found in this ergot alkaloid pathway, and we were wondering whether it is just a coincidence or it actually tells something. And we were also wondering whether it exists in other metabolic pathway, and what are the mechanism behind this. And Gillian probably can tell us more about this, whether it's, sorry. Oh, it's almost ready, right? Sorry. Okay. And okay, I'm almost done. And so, yeah, this is the last slide. As I said before, the ergot, the LPSA gene is super long, and we can't grab the whole sequence. So that's why we did nanoper sequence, and we include for a few strands, include one representative from each of the chemotype. And Tom, also from Dave's lab, he used this pipeline and grabbed the whole cluster from these strands and did the comparison. And through the comparison, he found the higher levels of polymorphism around the LPSA regions. You can see the red lines are more elevated than other places. And also, he also observed a stop codon at the second C domain of class 1, the string of class 1, and which might indicate for the class 1 strains might have only one functioning of LPSA. And another observation is the variation here at the second domain of A domain of LPSA A1, and the sequences predicted encode leucine or other branched hydrophobic amino acid instead of phenylalanine, which is other species, other strains produced. And this is actually, this observation talk nicely to the ergot alkaloid, the separation of these two classes, as we indicated earlier in that graph, class 1 and class 2. And class 1 had this branched amino acid in the second domain, but class 2 had the phenylalanine. And so another follow-up question would be whether this is only this sequence variation, whether it's only in this one single strand or also in other strands. And he went back to our Illumina sequence and only extracted this region, only extracted this from. So he was able to extract that region and did alignment and phylogeny. And based on this phylogeny and the DNA sequence analysis, other strands in this class A also had the same feature and the same sequence variation encode leucine and branched hydrophobic. So they collectively, based on this evidence, the variation sequence variation at the second domain or A domain of LPSA1, and also possibly due to the lack of the second copy of LPSA, might be the trigger of the separation of the two classes. Yeah, that's all I have to say. I just want to reiterate the few points the take-home message from each of the topics. For Clavis, taxonomy of Clavis, in pre-molecular era, the species were described by morphology of sclerosia, stomata, Canadia, sugar content, and EA production. And there were 67 named species, and they need to re-evaluate it based on molecular taxonomy. And for the molecular era, And Claviceps, the genus, was separated into 4 sections, and Claviceps propria sensulato was separated into several phylogenetic species, and the molecular-based monograph is needed. For the populations between Canada and the West US, there's three subpopulations. However, the subdivision is not associated with the geographic or host separation, and we don't know the reason yet. And so in terms of the variation of ergot alkaloid production and yeast genes, the variation in yeast production exists between sections, species, and strains, and which are associated with present absence of yeast genes, and also the variation in DNA sequences. So yes, and without all this help from our friends and all these people, we can't have any work done. So I think I just owe a big thank to all of them on the list. And thank you for your attention. I hope I didn't overtime too much.
That was great. Thank you very much, Mindy.
Thank you.
So great to have you sort of here. I wish you were really here. I'll open the floor for questions in just a second, but I want to say that we'll post in the chat right now a meeting room for a 1:45 meeting with Mindy and I and some other people, right, Mindy?
Oh yeah, okay, yeah.
Right. So
yeah.
1:45, I'll post a new Zoom link and you're welcome to hop on. Just
okay.
check for it in the chat.
That's great.
Meanwhile,
yeah.
Are there any questions for Mindy today?
Mindy, thank
yes.
you. Remind me, do any of the alkaloids have effects on plants?
Has you mean ergot? You mean the ergot on plant? You know, the sexual reproduction is certainly predicted, right? Predicted in the ergot, but whether they have sex on the plant, that's a question. Probably the sexual initiation is already there, and when they drop on the ground and they germinate and produce, segregate. I would think maybe that's the case. But another piece of information, I don't know whether I answered the question, but another piece of information is Tuzisky, and they had a lot of classic genetic study on this ergot, and they found even though heterozygosity is not necessary for the reproduction of ergot, but they found that, but it exists. It's not necessarily, homozygous is also okay, and they also observed, and it can also produce ergot, repeat the life cycle, but heterozygous also happen, also present. And another information, I don't know, another information is also very interesting. Very interesting is for the cubensis. Cubensis is one of the species I described, and we collected a few specimens, and one of the specimen and our first isolation is cubensis. And when we second time when we isolate again, because that culture kind of had some accident, we lost that culture, and then I asked we go back to the ergot again to separate, and we just never get cubensis again. We always get propria. So there is an indication, probably it's just a small segregation, small section maybe of the new genotype. And it happened the first time we isolated it, and afterwards we just never get it again. But which also indicate on that ergot there might be a hybrids even sexual reproduction in that, and also even maybe hybrids between species. I'm wondering. I don't know.
Hey, Mindy,
Any comments?
this is Gary. Could you remind me the graduate student study I think that you talked about, where they took a number of individual isolates coming from different hosts, but they were all in the Purpurea sensu stricto population, if I remember right?
Yes,
so
that's right.
Was there much differential specificity, virulence on the different hosts? I know they came from different hosts, but when they inoculate onto those 4 or 5 grass species, was there really much differentiation of the population that way?
I found I will just speculate based
Sure,
on, but if you want to know precise, better I will contact Jim Manzi. You know Jim Manzi, right?
Oh,
Do you know
yes.
him? Yeah, you
Oh,
know
yes.
him. Yeah, he probably can provide more information. Oh yes, it reminded me they had recently had a new publication and studied the host specificity and pathogenicity, different level. So there is different levels of pathogenicity, but I'm not, but it may not necessarily sure for each of those strains.
Right. But it's the general message that most of these isolates in the sensu stricto group are kind of generalists in their host range.
Yes.
Okay.
Yes. In
That's
general,
the thing that interests me the most. But another little thing, Mindy, I might just mention to you, we've had a chance in the last few years to interact with folks in the Alltech company. They do a lot of extensive mycotoxin screening. I think they screen for some 40-some mycotoxins in grains and whatnot. But one interesting thing, the last couple of years, they've been showing ergot alkaloids showing up in corn silage, which I find interesting because, as far as I know, corn is not a host for a claviceps. But my hypothesis is that, when you cut corn for silage, you're getting pretty close to the ground, and that they're probably harvesting weedy grasses that have ergot that are just growing between the rows of corn. But I don't know, would you have a different reaction to that finding, or?
That's very, very interesting. I was looking for the ergot on corns for a while, but I never found it. But I know there is a species. There is a species on corn. Let me check. What's her name? Let me check. Maybe I don't have it here because, there is a one. I think it's a gigantea.
Wow. I've never seen it. That's really fascinating. But
yeah, it's very fascinating and gigantea, and also people told me. I remember once I had a conference with another lady, and I said I study ergot, and she said she found corn in Canada, even in Canada. And I, during the summer, I try to find them. I can't. But and in some cases, I found it's different to tell whether, early stage, whether they are smart, whether they are smart or ergot. I don't know. Yeah, so there is a species, but it's very interesting, and maybe they can trace back to the field.
yeah.
You found your colleague found in the xylem, right? Maybe we can trace back to the field and see, can find some specimens of.
Thank you very much. Enjoyed your seminar.
You are very welcome. Thanks. I hope my talk is not too overwhelming.
Well, it was overwhelming, but in a very good way, I think.
Thanks. Thank you.
It's such an interesting system. And I have one question for you about, I think you mentioned when you were talking about the honeydew, that it varies in its sugar content, but it also varies in its alkaloid content. And I was curious about that. if I have that right, I think of honeydew as the way the fungus gets its asexual spores dispersed by bugs.
Yeah.
So why put alkaloids in there? Doesn't that mess up the bug?
Oh, you're right, Kathy. Actually, some recent study work indicated in the honeydew, there's no alkaloids.
Okay.
Very, 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 very low. Very, very low. I think I will say no alkaloids. And there was also a publication by the Canadian Green Commission. Canadian Green Commission, there was a group of scientists working on the alkaloid, and they had a recent publication compared, they even actually compared the honeydew and also but in culture, in honeydew, no, but in culture, sometimes some strains can produce.
Okay.
And that is also the way the pharmaceutical company, they want it, right? They kind of use different method, different way to stimulate the fungus to produce alkaloids.
In my head, I think of the alkaloids as a sort of protection for the sclerotium. Is that how you think of it?
I think so too. I think so too. I think, I always saw there is publication, but there are two publications recently, they indicated, from our perspective, we think ergot is a pathogen because they are bad for us and bad for the agriculture. And in a way, and for some species, they had certain levels of pathogenicity and can lower the production, the yield. But in most cases, actually, it can be called mutualism. They actually, one thing, as you said, the alkaloid actually protect the fungus from devour of animals or the insects or that stuff. Another way is the black color. They kind of scare away other people, the black, they did research actually, the black color actually also takes away their enemies. So in that sense, ergot actually is a mutualism, should be.
Okay.
I would say that.
Well, what about, so there's one species you mentioned, maybe Maximus, that you didn't find any toxin genes in?
Oh, yes, that's right. That's right.
What's that one got going on?
Yeah, that's right. We don't find any, but I honestly, I don't have a lot of experience with that species though. And I think those are like a tropical from from South Africa. Don't have a lot of experience. Yeah, we don't find any alkaloid and no genes. It would be very interesting, yeah, to look into it. It might be, but yeah, I'm wondering whether it's actually not ergot. but I think the Patrova paper did indicate other genes group it inside of it in the section. Yeah.
Well, are there other questions today for Mindy? Otherwise, I'll remind you that we're meeting again with Mindy at 1:45, and you're welcome to just join our Zoom group for a chat with her informally. And I just want to end, Mindy, by saying, I'm sorry it took us so long to invite you back, but it sure is nice to see you.
Thank you so much.
See you in person sometime.
Yes, sometime. Thank you so much for inviting me. It's great. It's great, even though it's not a person to person, this is also great. Yeah. Thanks. Thank you all. Thank you all for participating.
This has been a production of Cornell University, on the web at cornell.edu.